Good morning, good evening, everyone. So I'm Pedro. Here I will present the I present the Team 1 Sprint 1 summary. Okay, it will be like 6 to 7 minutes. So yeah, this is our team. It's a large team, many people from many different institutes. So with the main background here is statistics and plant breeding, of course. Yeah, so you can see from many different institutes. So what? The main thing the first thing we did was do a kind of overview and some demos on the current tools we are using. So each one try to show the softwares they are using, the designs they are using, their programs and institutes, right? So we did a couple of demos, and that was really interesting to see and to share some experience between us. With that, we tried to write a list. Let's say this, have a list with the most used designs and some descriptions of these designs, some details on it, and exactly how much we are using each design. And this discussion was really interesting because, as you saw, our team is a large team with many different people, each one with their, let's say, they do their daily things separated from other institutes. So that was a good discussion to share some ideas. Sometimes some designs are used with different names between different institutes. So it was good to try to, a good moment to try to standardize some ideas on this discussion. Sometimes it's quite complicated, right, to discuss the different designs. And after this, we discuss also the main tools we are working on and how those designs are, or the status of these designs, the experience designs in each of these tools here, right? And this was useful to show us some opportunities. For example, some designs that are very used or often used by the institutes that are not implemented in the tools we are using. And also we have always this possibility here. There is some designs that we didn't discuss, but maybe of interest in some groups, right? Here we create like a summary table to show the frequency or how much we are using each design. This here is just the IDs. We have listed many designs, and this number is just the IDs of the designs and the name we are using. And here you can see the different institutes and the crops, right? And the say the frequency that each crop and institute is using each of these designs. And we finalize this table here, just trying to at least here the most used, right? Some of the most used designs, and the status of each one of them in the tools we are developing or using, right? So the green one are those that we have already, and the white ones are those that we still need to work to implement in the tools. Okay, so what? With all this discussion, we could have a good understanding about the current state, compare and contrast experiment designs that are available for implementation. So it's important information, right, to start to work on all this. And of course, this is the opportunity we have. So if we having this good discussion about the different designs and have these designs available in our softwares and applications, for sure the idea is to have this increase of occurrence of increase in the occurrence of evaluation and the rate of genetic gain, since we have better designs, right, of our experiments. I have a message here in the chat. Let's check. Okay. Yeah. So now, for next steps, we have this. We have listed some designs that we need to work on, right? So now we are in the stage to work on the IT and to implement these designs. Have some discussion on this yet, right? But yeah, the idea is work on the implementation of this. And also some designs that we can discuss a little bit more, investigate a little bit more. Because when you talk about automated designs, we realize that there are many, it's a very, let's say, dense subject. So we have to discuss more this because we have many variants of this. And as we know, we have a lot of optimized designs nowadays. So we have a lot of research on that. Every day we have new things on this being yeah, available in some papers. So yeah, we need to also consider this in our softwares. Yeah, so is this? I hope that it was a good summary for all you, and in a few minutes you can work with the questions and answers. Right? Let's start. It's better to do this after the other presentation, right?
We have time for one question. Feel free to put in the chat. So one quick question, if you are ready. Okay.
Okay, if you have a question, we'll be here ready to answer.
I guess I'll just ask one quick question if we have time. So it looks, within centers that there's some standardization across centers. There's a lot of different designs used. A lot of that may be driven by the crop and how the testing's done. But is there going to be, are there going to be, workshops or attempts to try and across the various stages, do some more standardization of what's being done? I'm just curious of the relative value of kind of exploring designs versus sort of implementing new designs within EBS and where that kind of sweet spot is.
Yeah. I can say something maybe. So this is something I think Eduardo may have started to sort of socialize this a little bit. But as most of you have probably been involved in the past year in the concerted effort to provide genetic gain estimates and realized genetic gain estimates in a standardized manner to the funders in a report. And it was a huge achievement. It was the first time that the funders got sort of a system-wide report of genetic gain as a KPI. And we learned a lot about and made a lot of connections between biometricians at different centers and also breeders and biometricians in the course of doing this. And the mode of working that we figured out, I think, has tremendous value, and we didn't want to lose momentum in this sort of a more harmonization effort. So in the CTEH meetings, the breeding leads and Michael and the funders have been discussing in the next year, how do we build on this momentum? So the obvious next step is the genetic gain estimates. Some of the things that we learned from doing the genetic gain estimates has to do with the pitfalls of not having standardization in trial designs. Like not everyone, it was sort of a common thing to not have a good check strategy that over time, for instance, or and things like that. So over the next year, I think what we're going to try to do is to build on that effort to identify not just like retrospectively getting realized genetic gains, but start going prospectively, how we'll be able to do experimental design and run trials and gather data in such a way that that we will be able to provide better KPIs, yeah, to ourselves and also to the funders. So yeah.
Okay, thank you, Pedro, Kelly, and also Yonghua for that question. So definitely there's momentum to build for the experiment design space. And also we do have our industry consultant, technical advisors. And then I think within EIB, we have the gems of from Dow or Monsanto or Bayer and Captiva, Syngenta, all these big multinational companies, they have been doing the stage 1 optimization for experiment designs. So I highly encourage Pedro, you take on like Stephen and whoever basically you know in our space, Salvador and Brian. I don't know if Brian's on the line. So definitely we have Yonghua, Kelly, and then Eduardo. So huge industry expertise in optimizing the stage 1 trial design. So anything from our center where you identified as gaps versus best experimental designs can connect for better quality selection across year, yeah. So follow up on that for Q1. Okay. So with that, let's switch to the second topic for the sprint 1 in the BrIN 2 advancement decisions, which was led by Kelly. So Kelly, do you want to share screen? If you are ready, feel free.
I was muted. There we go. All right. So
Kelly, excuse me, you're in presentation mode. We see 2 slides.
okay. Let me, is that good?
Yes.
All right. So so in group 2, we were, we talked a little bit about analytics early on, but the core focus of group 2 is really looking at decision support. And when we talk about decision support, it's we run lots of analyses, we get lots of information back, but how do we process that information in a way that enables good decisions to be made relatively rapidly. One thing I was really impressed about with just BrIN in general was just the level of participation we had, how active the participants were. I mean, it really was great to see, because I think especially, as we get out into things like analytics and things like decision support, right? It's much more difficult to define a set of kind of core deliverables that are going to meet the needs for everyone. There's likely going to be lots of customization and personalization of how in particular decision support's done. So we really do need to have an active community contributing to the development of these tools, and it's great to see that momentum is there, and we have that type of participation in the BrIN group. So what did we look at? Well, one of the things we did initially was just to examine the available software tools for analysis and visualization. One thing that came through in the presentations that we had from members of this community was that a lot of centers have developed R scripts and R Markdown to really sort of check off the boxes of what you need to do an actual advancement meeting, right? So under excellence in breeding, we've tried to shift things towards having these advancement meetings where we have expertise from different groups in the room to make the best decisions. And I really thought, looking at what was done, at Erie, at ICRISAT, at CIAT with Mr. Bean, there are a lot of great tools out there that are being done. I think, Mr. Bean's broadly enabled, but, a lot of these R scripts do still require a lot of kind of manual manipulation of data. So I think there's room to improve and further automate some of these processes. But I think in terms of, getting at sort of the core of what's needed, there's been a lot of great work here. Those scripts have been shared. And so, we do have lots of information to work on moving forward in terms of what we can do in this space. We did a review of the advancement decision processes used by members of the BrIN community. Again, this was a mix of reports generated via R and, some really big Excel files that were color coded and sorted that are being used to make advancements, These reports, I think, are going to be a very valuable source of information on the types of data that we need to provide in these advancements. But it was quite clear that there is a big gap in visualization capabilities and automation of the report generation. I was kind of blown away by just how many columns some of these files had, and how difficult it would be for really anyone to kind of sort their way through that and identify kind of optimal advancements. So I do think there's real opportunity for some substantial improvement in this area. We also introduced the group to Spotfire and granted some licenses to some volunteer members that I'm quite grateful for sort of dedicating the time to start to learn that tool and to begin mocking up visualizations for us to start to look at and share. That was really happening towards the end of Sprint 1. So we didn't get to any real sort of finalized versions of these mock-ups, but certainly there was progress being made there towards the end. Just in multiple topics, it seemed like there were multiple ways in which people could get to having the statistics they needed. Granted that process was probably a little bit more cumbersome than it needs to be, but it seemed like most breeders had access to good analysis tools. But interpreting the many statistics produced by these pipelines still seems like a really big pain point to a lot of breeders, and something that I think should be a priority to address. So decision support and data visualization is recognized as a key gap. There are tools available, and Markdown seems to be, so R in general and R Markdown does seem to be pretty widely used at this point. And something to keep in mind, I think, as we look at how we want to tackle this space moving forward. And again, community members were given to Spotfire. I don't think it's clear at this point what the actual technical solution is going to be moving forward. But regardless of whether or not it ends up being Spotfire, I do think Spotfire is kind of a nice tool to be able to quickly spin up some dashboards. I think if we can tackle this problem, we are going to enable faster, more effective breeding decisions to be made. And I think that can have really huge impacts, especially in crops where breeders have to balance across multiple factors when doing these advancements, and multiple geographies in some cases as well. So now what? What do we want to do moving forward? Again, in this Sprint 1 was largely focused on gathering information, which I think we accomplished. We have, I think, lots of information that can provide a good basis for developers working in this space. I think the next step is, this needs to be effectively communicated with software developers. I think this is happening. I think there does have to be, people working in this space that can kind of translate between the users and the software developers to make sure nothing's kind of lost in translation there. The community has developed some solutions, or at least partial solutions. And I do think that again, when we get into decision support, I think because it is a space that's going to require lots of customization, I think we need to think about tackling this in a way that supports the community and how they develop innovative solutions to be able to handle decision support. So, recommendations from the breeding programs. one thing is, we need to make sure needs are clearly communicated to developers in this space. I think that this BrIN community has been a great way to really understand what's happening and to get information directly from the users. Again, I haven't been as involved in that sort of process after gathering this information, but clearly that there needs to be a very effective way to move this to the developers and to be able to actually define specific use cases to start to tackle some of these challenges in the space. I do think, in decision support, providing mock-ups can be a great way to facilitate this communication. I do think there's, especially with the tool like Spotfire, even if it's not a permanent solution, it does provide a great way to have very sort of interactive development of decision support tools between the dev teams and the users. And I would really love to see this community move forward and really start to serve as a place where the dev teams and the users can come together and really talk through some of what's being done in the space for EBS. This last point is probably a little bit more personal opinion than anything that came out of the GRIN group, but I do think, again, that software solutions in this space do need to provide the community opportunities to be innovative. I think looking at strategies that can allow users to take what EBS delivers, but also be able to make it work in the way that it needs to work for their crops and their breeding program objectives. And so that's all I had. Thanks for giving me the time to talk today, Star.
Thank you very much, Kelly. Because of time, and also there's a great segue. Let me share the recording because Xiaofei has another meeting, and he recorded representing the second sprint on stage 1 advancement. As you all know, team 1 and 2 excellent design advancement converged into one team in the second sprint, and Xiaofei representing the second group first recorded this, and after that we can have a few people share who are willing to share. So let me share my screen.
I'm Xiaofei Zhang, the castle breeder and CS. So today,
can you hear?
I can, yeah.
Yes, you can hear. All right, so let me start.
Today I'm going to summarize the discussions we had during Sprint 2 about stage 1 trial advancement. So we decided to use stage 1 advancement as an example to help us understand the data flow and how breeders analyze the data. So we divided the team into 3 groups. Each group was led by breeders, and also we got a lot of input from statisticians and database scientists. So we covered the major crops in CGIAR, and we have the scientists from 3 databases, and there are different tools available for the analysis. So
quickly, just very, yeah. So Erie Accuracy led by Abhishek and Harish, and there's a large team from Asia. And then team 2, there are more people in this team as well. And the Xiaofei only have limited information. Only the 3rd team captured maybe the entire team. So just an update here.
For each group, we have separated discussions about stage 1 trial advancement, and then we sit together to put our discussions together. So we all agree this data flow. So first we access the database, and then we do correlation, double check several items, and then we clean the data and using basic analysis, identify outliers, missing data, calculate the heritability, and then we do trial analysis to calculate the blob, selection index, make selection, and using the result from analysis, we visualize the data and bring the figures, the table to advancement meeting that can be understandable by broad audience. And there are they already have some tools available to analyze the data in each step, and there are also tools integrated in database. However, there is no individual software or database can handle the whole data flow. So during the discussion, the database realized this gap. So database reached out to the developer of Mr. Bean. We have several meetings to discuss how we can build the connection. So finally, the developer Johan developed some feature in Mr. Bean through BrIN, and now so Mr. Bean can read in the data from a cassette base. Mr. Bean also can read the data from BMS. So once we build that connection, then we can use Mr. Bean to analyze the data in BrIN base for the basic analysis and standard yield trial analysis. But we still have to have gaps in data curation and data visualization. I'm going to show some details. So here for data curation, we agree that at least there are 4 items we need to double check. First is the trial layout. So that's about the row, column, and information and the reps. So we need to use this information to fill the linear mix model. So we need to make sure all this information is correct. Another point is the clone name or the line name. So there is breeder's name, there is release writer name, and when we share the material with partners, they might give them a different name. So we need to make sure in our analysis, the same genotype should have one name. And about the unit and about the missing value, so sometimes there might be some error there. We always need to double check before we analyze the data. And if we find the error, we need to be able to make changes in database. So I'm thinking this part, probably no database can handle all these items, and especially can come back to make changes. So we still need to keep working on this part. So another part is data visualization. So we have our scripts available and we have softwares available, but they are not integrated in a software or in a database. So finally, I'm going to summarize our discussions and achievements. So first, we all agree about this data flow and the analysis in different steps. And we build the connection between Mr. B and the BrIN base. So we still have gaps in data curation to double check the different items before data analysis. And also we need to integrate tools to visualize our result. So I think these two points might be something we can continue working on during the Sprint 3. And really, I'm looking forward to the discussions in 2022. Thank you.
Yeah, thanks very much to Xiaofei and the entire stage 1 analytic team with all the compare and contrast, consolidating different crops into this final summary. So anyone from team 1 or 2 in Sprint 2, you want to share, I think just 30 seconds, that's all the time we have. You can unmute yourself before we move on to the next presentation. That will be from BrIN 3 GIS application. So Roberto, so get ready to share your screen while we invite a couple of people to share. Okay, so Kelly, please unmute yourself if you.
Sorry, I just had a question. I don't know if there's anyone from that team on the line, but so when we reviewed Mr. Bean, there were certainly gaps in analysis. Perhaps that wasn't for stage 1 analysis or early stage analysis. Maybe that did have that completely covered. But do we feel like Mr. Bean is a fairly complete solution, or were there gaps identified by the group that Mr. Bean doesn't meet in the actual analysis space? Obviously, in the data cleaning and the visualization, there were some gaps identified.
Johan, are you on the line? You can unmute. I don't see if Johan's on the line. He has to jump between meetings as well. Anyone else?
Looks like Abby has his hand up.
In terms of answering Kelly's question in Mr. Bean's compare and contrast, Pedro, do you remember in the compare and contrast, the feed gap analysis? Bird? Okay, so Kelly, we'll have Johan to answer your question. So very big Excel file in the feed gap analysis in all the standalone tools, including Mr. Bean.
Yeah, and I could always follow up with Jaffe and like an email or something. So thanks.
Yeah, sure. Abby, please, 30 seconds, please unmute. Thanks.
Yeah, thank you, sir. So yeah, regarding to like all of this is sprint, I think Kelly also raised it regarding Mr. Bean. So Kelly, I think different tools, methodologies, technologies were tried, and it was overwhelming to see like people have different ideas. I think a couple of readers have shown very nice, stunning visualization from Spotfire as well as from R-Shiny. So I think lot is still, I think we discussed IN star and what we discussed. There's a lot more probably need to synthesize and come in a form. But I think everybody has contributed, and the most important part, the workflow is very clear to I think everybody. The whole the workflow, how things need to move forward, that's quite clear.
Sure, thanks, Abby. Anyone else from team 1 and 2 want to share? Okay, I don't see hands up. So Roberto, you're ready to play your recording, or you can share your screen.
Start. Could you play from your PC?
Okay, sure, I can. Yes.
Thank you.
Yeah. So next, we are going to transit to the genomic selection application team.
Hi everyone, my name is Roberto. I'm a senior scientist at IRI, and for today, I'm going to present some results that we have obtained over this year in Group 3 GS applications. One of our major concerns is to identify commonalities difference and the next steps to fully implement genome prediction in our centers in a 1 CG scope, not all, for all centers and all breeding programs. So over the year, we have mapped the current states across breeding programs and also the centers. Regarding practical and analytical procedures, so we collect information from what's the best stage to apply genome prediction, the population size, the training set compositions, and so on. But the main focus for today is to present the commonalities and difference for the analytical procedures or the analytical pipelines. So to collect this data, we hand out 2 surveys, and we could capture more than 42 aspects, topics grouped in 6 sections, from training set, phenotypic quality control, until breeding applications. Just to highlight the representativeness of these surveys, we could capture the information and the opinion from 19 breeders or biometricians, 6 different centers, 2 NARs, and 5 crops, covering the most important crops and also the 3 different reproductive systems. So talking about the genome selection commonalities, 3 of these sections are more related to the analytical pipeline, the main focus for this presentation: the phenotype quality control, genotyping quality control, and prediction models. The first is already implemented on DBS for the phenotype quality control, but we need to define some standardizations in terms of procedures. Just to have an idea about the topics, it's if the breeders or the biometricians adjust the phenotypes for year effect using checks or study checks, if they remove outliers, transfer to normality, or spatial analysis. Most of them are already implemented on EBS, but there is no specific threshold or like recommendation for that. So that's one thing that we need to get together, biometricians and breeder, and try to define these procedures. For the last 2 topics, type in quality control and prediction models. The good news that there are more commonalities than differences, and it's amazing because it will help us to unify the analytical framework in the easiest way. So talking these commonalities are mainly for these the two last topics. Some of them are almost standardized in terms of QC parameters, thresholds, and so on. And also they are available in other platforms for downstream analysis, or at least for this upstream analysis, such as Gobi or Galaxy, but not on EBS. To fully implement on EBS and make it available for everyone across centers, there are some challenges, mainly for the read depth and haplotypes in market dosage for polyploids. But for diploids, or for those crops that are not so interested in these specific ways to model genome prediction, the pipelines are almost ready. In terms of R or any other platform for coding and script, creating these analytical pipelines. Just to have an idea about the topics that you asked breeders and biometricians, the way they eliminate markers based on missing data, the allele frequency if they input data, or if they prune the markers based on LD until dosage or read depth. Concerning prediction models, what's the method that they use for cross-validation, kinship, the way that they analyze include GBI models, and also if they use or not a different procedure or way to model residuals. Unfortunately, just one topic is under test on EBS for the kinship one after the post-quality control. But yeah, as I said, the good news, the scripts are ready. There are good consensus in terms of parameters and thresholds, so it will help the EBS team to implement these analytical pipelines. So talking about like the bottlenecks and next step, overall the analytical pipelines are similar already. As I said, it's a really nice it's a good news. However, we have some problems, and these issues resides in combine these fragment data layers for downstream analysis. We have phenotypic, genotypic, and pedigree data, but we need to smooth the script or make it easier to combine this data for downstream analysis, and also spend more time to define quality control procedures and for phenotypic data. Finally, we believe these results and these findings will help BrIN, Group 3 and Group 4 and EBS teams to develop and implement tools to support breeders' decision, mainly for genome prediction implementation. I'd like to thank you, everyone, and the team 3 for all the support, breeders and biometricians for the collaborations.
All right, so Roberto's here. So not only you can ask the question, you will have answer here too.
Great, thank you, Star. Thank you for playing.
Okay, so we have time for a couple questions. Okay, let's see in the chat. All right, any questions for Roberto, genomic selection application team? Please unmute yourself.
Maybe just one question. There was a lot of focus on like the mechanics of doing genomic predictions. Did you get a sense of how many programs are either using genomic predictions or are planning to use genomic predictions in the near future? Or is that not really covered?
Yeah, that's a really good point, Kelly. Yeah, we cover, as like I described in the beginning of the presentation, we collect the data, information from 6 different aspects for genome prediction, and 3 of them are more related to the practical part. But for this presentation, I focus on the analytical, the framework, the analysis. But yeah, we have different, like many breeders in different stages, and some of them like in the beginning of the implementation, and some of them like really like applying genome prediction. And this variation is not more related to centers. For instance, in IRRI, we have 1 or 2 pipelines that are really implementing genome prediction this year or in the coming one, but we have some programs that are not ready yet for that. So this variation is not a specific or center or crop. Depending on the breeder, there are some variations, but yeah, we can see different stages across centers, programs, and within centers.
Sorry, just one follow-up question. So if we think about genomic prediction using like GBLUP, and then running, just genetic evaluations for parental selection using, relationship matrices generated from pedigrees and markers, the mechanics would be somewhat similar. Is there a need in the genetic evaluation space as well, or was that sort of treated as being distinct and separate from what your group was covering?
No, I don't think it's not like distinct. So I can see like a huge like difference in terms of yeah, as you said, it's almost the same mechanics, but there are many other aspects that during like for group 3, it's just like one part of the activities that we covered over this year. We have many other things that for instance, part 1 present and discuss some issues regarding logistics to fully implement that. So I think that's the main reason that we have like one specific team covering these aspects, because we need genotyping, like, or send the samples or the entries to the NARS and receive the data back. So yeah, that's the reason. But regarding the analytical part, so yeah, I can't see any like reason for any specific group for that. So it's almost the same mechanics. However, there are some demands, some logistics and so on that are more related to that we depend to fully implement genome prediction.
Thanks.
Yeah, as Roberto mentioned, 19 participants entered compare and contrast across different crops centers in terms of their GS pipeline from all the way those 6 sections. So now I invite whoever participated in this survey, if you want to share yeah, what, so what, now what, anchoring with what Roberto mentioned, feel free. We have maybe 30 seconds, that's it for each person if you want to unmute before we move on to team 4. That's molecular breeding and QC, that's what they focus on. So before we move on there, anyone want to share? You can unmute yourself. All right, okay. So we have the, okay, go ahead. Okay, if not, let's move on to team 4. That's the molecular breeding group, and they focused on quality control. So Damien, Mokhtar, Cesar, who do you want to kick out first? Maybe the upstream. So we divided upstream QC and downstream QC. How about we start with upstream? So Cesar, feel free to share your screen.
Yeah, I can start. Just let me share my screen. Can you see my screen?
Yes.
Okay, excellent. So hello everyone. Well, my name is Cesar Petroli. I am scientist at CIMMYT, based in Mexico, working part-time on this project where we are trying to define the marker-based breeding analytics, development, and implementation in Group 4. Today, I'm going to present some of the methods and tools that we are using to develop a framework to achieve this objective. And well, as you know, if we will work with molecular markers, we need a space to store and track the good quality of the genotypic data. And our idea is to use the enterprise breeding system, where it will be the space where user can, for example, require some different services. But today we will more focus on the genotyping service request. The objective of this system is to generate an efficient pipeline that could host and support the creation of genotyping service requirement for many different and multiple research institutions. So the service manager is connected and supporting and is supported by 6 components. The core system, where the user is going to take or push the samples, the request manager, the sample manager catalogs, data manager, and the vendor manager. Here, as you can see, the genotypic service request, it has multiple functions. Of course, it has the ability to create new requests. Also, it has to create budgets and prepare shipments to send to the vendors. And of course, it will receive the data and storage the data, retrieve it, and download all the genotypic data created or produced by the vendor from database, according, of course, with the user requirements. Well, we had some, of course, some advantages. The main one is that the idea is to create an automated process, and well, it has to be also friendly and accurate. In terms of limitations, the main one is consensus on the user needs. Every breeder has different needs, so we had to attend those needs, and it's quite difficult to.
Cesar, are you there? Or just myself? I couldn't hear you anymore.
This manager dashboard looks like, with the menu on the left side, which each steps that have to be followed in order to create a new request. You can see here on the left side. Of course, we can make updates about any genotyping request here, which is, or what is the next step? Well, at the beginning of next year, we want to make an update with the new features to users or molecular breeders and breeders, improve what is already done, and of course, increase the service catalog with more services. So in group 4, in this first sprint, we were working on the Upstream Data Qc, using the integrated system based on the EBS pipeline and the Gobi database. The Upstream Data Qc has 2 steps, 2 main steps, the pre-data production and the post-data production. In the first one, the idea is that the sample manager will share the metadata information from each service request or typing service request. The sample managers, as I said, will share that data or that information with Gobi database. And of course, must match later when the data manager receives the data files from vendor. And the second one is more about the details on the content of the data files that the data manager receives from vendor. That includes, for example, we will get, or the data manager will get the expected number and name of samples, the expected number of and name of markers, expected data formats, missing data to have the same codes always. And in that aspect, we are working right now with vendors. specifically where we are right now in this work. Well, EBS is done. The genotyping data request is almost done also. Next year, we'll start with the technical aspects on samples and data tracking with Bobby team and the online job submission with vendors. And well, the question here is how the data will be extracted from database. Well, also we have two aspects here to lead with. One is the technical challenge that we expect to solve it with our Gobi friends. And the other is the concept of the objective of the data displayed, which will be more addressed, of course, on the user needs. That's why in the second sprint, we were more focused on data display. The idea here is to create an analytic pipeline where the main functionality will be the merge of the data produced by different genotyping platforms and also different genotyping jobs. Try to avoid duplicate values, standardize the formats with maybe if needed, we can convert or make some conversion of those data. And the big question here is how we will deal with the unclear poor or poor quality data and how we will deal with the monomorphic data. You know, sometimes when in one population, the markers looks monomorphic, but for another population, the markers don't looks like a monomorphic. So here we use a template which has been kindly developed by Damian here. I think Damian's here. And colleagues and users from IRI and CIMIT, they has created this use case in order to complete the topics that you can see on the left side with the inputs. The inputs mean what the breeder wants or what kind of information or markers, list of germplants or list of markers they want to retrieve from the database. So we can make also some basic transformations when it's applied and displayed all this information in a simple table. The output of this module will be used as input for many of the upstream and downstream analysis. So in EBS and Gobi database, we are working to establish the necessary strategy to achieve these objectives. And that's the end. I would like to thank all the people involved in this work, EBS team, Gobi team, module 3 of EIB, and of course the vendors that can, they help us a lot with this. And of course, users and collaborators. And that's all. Thank you very much.
Thank you very much, Cesar. Yeah, given time, we won't have time to ask questions. So now let's switch to the downstream. Maktar or Damien, who want to go next?
I can take it away if you like.
Sure, go ahead, Damien.
Hopefully that is coming up for everybody. So this in sprint 2, we kind of split to two separate streams. So one stream was looking at the basic data display and data aggregation across different genotyping jobs. So that's what Cesar has just described. And that kind of forms the foundation or the basis for, you know, a lot of the other analytical pipelines that we will be dealing with in BrIN Group 4. And so one of these other pipelines, one of the first use cases that we decided to tackle was really looking at parental purity and F1 hybridity testing. So I'm just going to talk, you know, just very briefly on that. I realize we don't have a lot of time, so I will be going through this fairly quickly, as long as my computer will respond. Okay, so the use case here is in the breeding process, we need to determine whether our parents that we are using in the crossing process, for example,
are actually, what we want. So in other words, for an inbred crop like rice, are they pure? Are they pure? Are they the genotype that we actually want to use in our crossing block? And then the second use case as part of this is F1 hybridity testing. So we kind of lump these together because the analytical process for these is very similar. So with F1 hybridity testing, I mean, obviously it's basically to ensure that the progenies of our crossing process are actually true F1s, because I mean, even the best crossing process has a certain level of, for example, selfing of the female parent, or potentially outcrossing with non-target genotypes. So running the QC process on these two things makes sure we are crossing the right parents and getting the right F1 progeny that we are looking for. So the current situation is this process is mostly handled through kind of low throughput tools. I mean, we've got here a screenshot from Flapjack, for example, where we've got our samples here. These are our F1 samples in this case. These are the two parents of those F1 samples. And then we are using essentially just going through that and doing like almost like a manual inspection of that data to see which ones are true F1 and which one are not. Now, it is a fairly simple analysis. I mean, we've got our two parental genotypes here. We're basically just saying, okay, if these are our parents, what is our expected F1 genotype? And does each of these samples actually match that genotype or not? You can see there are issues here with missing data and potentially ambiguous data from time to time. So it's not all of the markers that are used are going to be informative. Obviously, a significant number of markers are also going to be monomorphic. So the analysis in any given cross is going to be restricted just to these markers that are actually polymorphic and informative. So the input data is the list of samples, obviously the germplasm that is being genotyped with our QC markers. Depending on the workflow, then it will be necessary to retrieve the parental information from the database. So for example, so if it's an F1 hybridity test, we need to retrieve from the database the parents of those F1 plants. And then the second input data is the genotype data matrix. So these lines have been genotyped with our purity or QC markers here. So we need to retrieve the genotype data not only for the actual F1 samples, for example, but also if it is an F1 hybridity test, we may need to retrieve the parental genotype data if that's not internal to the data set. So, obviously it's best if it is internal to the data set, but it's not essential. So the analytical process looks like this. Now, I won't go through that because I know we are short on time, but essentially, it can be reduced down to a decision tree workflow like this. But essentially, it goes through, QC test on the marker data. So, going back to what we saw before, basically saying, is this marker going to be a reliable one? Is it something that we should use in our analysis or not? And obviously also at the same point, we would be getting rid of or not considering monomorphic markers. For F1 hybridity, we will have to, as I say, identify the list of parental materials and retrieve the marker calls, not only for the F1 samples, but from the parental material. And then we have to calculate the expected genotype for the material. So for pure lines or parents, if there is prior information, then this would be the same as the parental genotypes of the QC markers. So we would be testing the current test samples against the previous gold standard genotype. If there is no prior information, though, we would have to calculate a consensus genotype across all of the markers and then compare each sample to that consensus. For F1 samples, we've already illustrated that. We retrieve the parental genotypes, calculate the expected heterozygous genotype, and then compare each sample to that. And so then, yeah, so this is really to what extent it is complicated. This is where the complications usually are. Then it's simply a matter of determining the match of the test samples with the expected genotype. If they are an exact match, then it is, for example, a true F1, or else it's false. So the output, again, I'm using the F1 hybridity here as an example. But for each sample that is genotyped, the result is simply either true or false. It is either a true F1 or it is false. Or if it was a hybridity, a parental purity test, it would either match the expected genotype or it would not match. You could, you know, as an extension of that, you could report the number of markers which are, you know, going towards informing this result. It's not necessarily a case of more markers is better, though. I mean, it might be a bit of a concern if you only have one marker that, you know, this result is based on. But other than that, you know, this is not a, how can I say it? It's not a competition to see how many markers we can have here. As long as we've got a modest number of informative, reliable markers, this is going to be what we need. So for future outlook, you know, one of the things, and I think this was already on one of our lists for one of the latest sprints for Group 4, but it would be really helpful if we could have a workflow to allow dynamic calculation of the optimal marker set based on the input list of germplasm. So for example, if we have a list of lines in the crossing block for a year, we can query the database based on, you know, previous results and say which QC markers or which other markers, even trait markers potentially, are going to give us the best discrimination amongst that set of parents there. So, you know, that would allow us to have a situation where we have many more, you know, actual informative markers in a given crossing block than if they were chosen by chance. So that's all. Thank you.
Thank you very much, Damien. Okay, given time, let's switch to Maktar. Maktar, you're ready to share your screen?
Yes, sir.
Thanks.
Just share. Can you see my screen?
Yes.
Yes, perfect. Thanks. I just want to share here like the potato QC marker panel and also like for germplasm identification. We've been working in the sprint too into mapping those different QC pipelines and also requirements for QC analysis. And just quickly, maybe I should start with the second slide actually, but anyways, I will just go quickly to what we have done. Like we started with cell cap markers and actually we had filtered markers from over 3,000 to end up with 24 markers. But then in the marker design, some were not validated. And I mean, long story short, just we ended up with now 21 functional markers that we use. Since last year, we are using routinely in the breeding program. And so we use them for like actually in several stages of the breeding program. It's because we are working with vegetatively propagated crops. So what we have from the observational trial should be the same clones that we use actually in the multi-environment trial. So it's very important for us to know that we are using the correct genotype at each stage of the breeding program. And also, since we cannot, like what Damian was showing, we cannot for now do hybrid purity with tetraploid. For the tetraploid genotypes, we are planning on using the QC markers, at least in the crossing block. So to make sure that at least we are using the right parents. And the process of how we do the QC is quite simple. Like we prepare either tuber samples or leaf samples that we ship directly to Intertech, and we get the results quite quickly. And that's really useful for the breeder to know when, like at the moment of selecting after the breeding, after the trials, to know exactly which genotype he would select, and that would help increasing the selection accuracy. And for the next steps, I will come back to it in more details. Just wanted to mention that for potato, we work also with tetraploid, and as you know, cusp markers from like what we get from Intertek are the deployed calls, but still, like there is a graphical, let's say, visualization of the data that can show the clustering of each ploidy level, but actually we don't have the results in the tetraploid level. So there is an additional step we do to have the markers at the tetraploid level. And as you can see here in the diploid level, where we have only one level of homozygosity, there are some individuals that are still clustered. This was done with 20 markers after removing one for missing data. And these were with 114 clones and were all full SIB or half SIB. So it means that it just shows us that the set of markers we have can work quite well. And you see here in the tetraploid calls with the same markers, we could actually separate all the individuals. Yeah, just a small explanation of what we do, and also the analysis workflow, which is pretty simple actually, because from the raw data we have developed a NAR script that takes in charge all these steps. So we just input the Intertek data, and we get directly the output, like the results from the QC analysis. So very quickly, sorry, we just remove the markers with a lot of missing data. Also, maybe this would not be necessary because any actually data point can be useful. The problem is, yeah, when the marker is not present in the reference sample. But yeah, I will not go into the details, but I will just quickly say that we remove markers with a lot of missing data, and afterwards we do the tetraploid calling. And to do the actual QC analysis, we just calculate the genetic distances between two samples, because we have the reference sample, what is supposed to be the correct genotype, and then the samples we want to test. And if there is a distance of 0.1, we just consider that it's an incorrect or a different genotype. And as I was saying, we do have a NAR script to upload and process all the data, and also to provide this sort of visualization of the results. I mean, the breeder receives an Excel file anyways, with true and false entries, but still we would provide also this graphical output where you have different environments here, and here is the reference and also the genetic distance between the same clone sampled in different environments. And I wanted to share with you the availability of our last publication on these QC markers, so you can visit the paper, and I would be very happy to answer the questions. And yeah, now what? So what we have advanced, but still there are some bottlenecks, and like linking what we're doing in the lab with the database, from the submission form to having also the raw data directly stored from the vendor into the database. That would allow less data being shared by email and then being uploaded. And even for long-term use, for example, now I'm receiving maybe the results for SIP, but in maybe 10 years, we would like to go back and see. So if we had the data centralized in the database, any user can actually see the data. And also, as I was showing before, we use a 0.1 level of similarity to declare a genotype different from the reference. But that is actually an arbitrary number. We do not have yet a very objective way to measure, because since the cusp mark analysis comes with a little, let's say, proportion of error, we do not want to, and also we do the tetraploid calling, so we did not want to be very strict into like how to define the level of similarity, and we do not know yet actually what is the best way to go. And also having the markers in the dark tag panel or developing QC markers from the dark tag panel also would be helpful to reduce costs for genotyping the reference samples. And also, I think I will just stop here with this point, which is for me very important. Like we do subsamples. We have subsamples of our trials. For example, we have a trial with 500 individuals in 2 replications and evaluated maybe in 3 environments. So we cannot do QC on all the plots or in all the plots. So having a kind of way to subsample the population, that would be very helpful to optimize the QC analysis, or at least to spot the environments where we have more, let's say, QC or more mislabeling. So that would help us actually maybe to focus on those environments where we think about QC. And yeah, the 2 next point would be just something the user for now won't be doing, like storing the information back into the database. So in the future, the same genotypic data can be used. And also that is not very a very high requirement, but it would be nice to have. So to have the extrapolate codes directly from the vendor. And yeah, I would be glad. I will be glad to receive your questions if you have any. And thank you very much.